Our first speaker will be Sven Sichon from Jülich, Germany, who will explain to you the structure, measurement, and analysis of genetic variation. Thanks. Is there a pointer?
Oh, yeah. Right. So good morning, everybody. So as Jason pointed out, thanks for the nice introduction. I'm going to talk about the Mac. Okay. So I'm going to talk about genetic variability, and what you should learn here is some fundamental knowledge. You should get a feel about the variability in the human genome. What are the most important types of genetic variation that we encounter? What are the possible functional consequences of genetic variation? And we will touch a little bit on the techniques, the genetic techniques, molecular genetic techniques and technologies to individually measure that genetic variation, because this is what we want to do if we want to test the influence of genetic variants on brain imaging traits. Now, it's been known for a long time that the genome is variable. So about 50 years ago, when people started to look at chromosomes under the microscope, there was some sort of variation visible already there. So for example, you may have pieces of a chromosome that are simply flipped, yeah, and that doesn't have any consequences in terms of a strong disease or so that you may develop. So clearly something that is sort of genetic variation. Or then you may have certain instances in the population where people have translocations, balanced translocation. That means that pieces of DNA between non-homologous chromosomes, between different chromosomes, are exchanged. There is no loss of genetic material, so these people look fine, yeah, but in fact the genome looks different than in other individuals. So that would also be at the chromosome level some sort of genetic variation. But since about 15 years, when the Human Genome Project was completed, we started systematically to look at many individuals in populations and try to find out what sort of genetic variation there is at the submicroscopic level. And obviously, that variation is huge. It's enormous. And we can classify that variation according to the size of the variation. So it starts with variability at the single base level, and then it gets bigger. So this year it involves from 2 bases to 1,000 bases, or then even larger than 1,000 bases, and then it goes to the microscopic, to the chromosomal level again. So, and we will touch on these most important types of genetic variation dependent on the size. Now let's start with the smallest yet most abundant form of genetic variation, the so-called single nucleotide polymorphisms, or SNPs. You will be very familiar with that name. So basically what it is you have a certain position in the human genome, and you can have 2 different bases at that position. In this case, for example, you can have either a C base there or a T base. And you can look at the chromosomes in the population, and you will see that the C allele is present on a certain fraction of the chromosome, and the other allele on another fraction of the chromosomes. It all is 100% in sum, and we call the more abundant allele the major allele, and the less abundant the minor allele. And this is just terminology, basic terminology. You will probably all know that, but it's important. So you know that we all have our chromosomes in double. We get 1 copy from our father, 1 from our mother. So each position is there in double. And if you look at a certain SNP position in the genome, you can have 2 different bases in your genome, then you are heterozygous. Or you can have the same alleles, then you are homozygous. Now, how many SNPs are there in the human genome? About 10 to 15 years ago, people have really systematically looked for the amount of SNPs that are in the human genome, and you can subdivide those SNPs according to their minor allele frequency. So usually everything that has a minor allele frequency of 1% or more is called a SNP. The ones that have a smaller minor allele frequency of less than 1% are called low-frequency variants, or even if they get rare variants. We will hear about that later. But SNPs, there are about 12 million in the human genome overall. And that was really, it was astonishing. Because that's quite a lot of variation in the human genome. And basically what people wanted to know is, do any of these SNPs have to do with, for example, brain imaging traits, or do they have to do with diseases? And in the beginning, people asked themselves, what do we have to do to systematically look for an influence of these millions of SNPs on these traits or diseases? And it was really frightening, because people thought in the beginning we have to test 12 million SNPs if we want to do that systematically. But there is something, and it's a particular thing about the structure of the human genome that helped researchers a little bit, and that has to do with a phenomenon that you all know. It's recombination. And so there is something like crossing over and recombination that happens during a certain process where the gametes are formed, so the egg cells or the sperm cells. You know, all cells have, I told you, all cells have a double set of chromosomes, and during that process, the number of chromosomes is reduced to a single chromosome set. And in that process, the homologous chromosomes will pair, and they will form these crossovers, and then they exchange material. So there is no loss of genetic information, no loss of base pairs during that process, but it leads to a new combination of the sequences that are on both homologous chromosomes. And what you get if you paint your homologous chromosomes in white and in black here, you will get these harlequin chromosomes. And from these chromosomes, from these recombined chromosomes, the gametes are formed. And that leads to a constant recombination of genetic information that is present on homologous chromosomes. So they are mixed all the time. Now, let's look at this across generations now. So this is a pedigree here with father and mother and 4 kids. And if we look at, let's say, chromosome 1, we can color these chromosomes because they all have different sets, combinations of SNP alleles, for example. So we can really distinguish these chromosomes 4 in the parent. And they are now transmitted according to Mendelian laws. So each child would get 1 chromosome from the mother and 1 from the father. And I have just made a cross here on that blue chromosome. That's, for instance, a particular position in the genome that is of interest to us. And let's see how this is transmitted across generations now. So what would happen is the children, they get 1 chromosome from father and mother. There are some recombined chromosomes, as you can see here. Yeah? And our position of interest is here. So two of the children get this position of interest. And now we go along over many generations, and what we will find after many generations is a set of recombined chromosomes, as you can see here. And what you can see is we have all these chromosomes. They are very colorful now due to recombination. But what we also see is that obviously the pieces of chromosomes that are recombined, they have almost the same size. So we have positions where obviously these recombinations took place, like here, and not in between. And now our position that is of interest, for example, it's still, of course, on the blue background chromosome. So it's on the background of other variants that are here in its vicinity. So this is basically a block of DNA material that is transmitted as such across generations. And recombinations are occurring at these positions, but not within the blocks. So here is a different version of that. So these are these so-called blocks. That means these are pieces of the chromosomes. They can contain many SNPs, but there will be, across generations, no or almost no recombinations within these blocks. And then you have the recombinational hotspots, like here, where all the recombinations occur. And that has consequences, because you can say within a block, the combinations of SNP alleles that you have, they are maintained across many generations. So they form certain haplotypes. You have fixed combinations of alleles within these blocks, and they make up a certain number of haplotypes, and the number of haplotypes is usually much smaller than the number of SNPs that you have within such a block. So in this case, you have 4 haplotypes, in a block that contains, I don't know, maybe 10 or even more SNPs. And there are usually a small number of SNPs that allow you to distinguish these haplotypes. So a lot of the information within these haplotypes, SNP information, is redundant. So you know the allele that is present at one SNP position, you can predict the allele at the next SNP on that particular haplotype block. So that means you have to genotype only one SNP, and you can predict a number of other SNPs within that block. And what you can do, if you pick your SNPs for genotyping in a smart way, you need to genotype a very small number, and you will know which haplotypes you have. You can identify the haplotypes that are present in the population by genotyping these particular SNPs, and they are called haplotype tagging SNPs. And this is a different view of that. So a larger part of the chromosome that forms a block, and you see there are quite a number of SNPs. Here is the sequence, the SNPs within, and here this is just the information of the different SNPs within that block. And as you can see, there are only 3 SNPs here that would really distinguish. If you know the allele at that particular SNP position, you know what haplotype you have in your individual. And these haplotypes, you want to test these haplotypes for association, for example, with your brain imaging phenotype or with diseases. And it sounds a bit trivial, but I told you in the beginning, when we knew that there are millions of SNPs in the human genome and we wanted to do association study, everybody was really scared because you cannot test millions of SNPs, or it's very laborsome and it costs a lot of money. So this reduction of genotyping work for systematic association analysis, that has obviously many advantages. It saves labor and it saves costs. So something that is important to know, these blocks, they can differ between populations. So if you have a block in the European population, for example, there can be 2 blocks in the same area in, let's say, African populations. And you have to know that when you do your association study and when you predefine, let's say, your haplotype tagging SNPs. And there are public projects that have large international projects that have defined these haplotype tagging SNPs. And one of these projects was the International HapMap project, and there is now the 1,000 Genomes project. The International HapMap project looked at SNPs only, and they made maps of haplotype tagging SNPs for different populations. And the 1,000 Genomes Project also includes rare variation. I mean, people are studying now more and more also rare variation, and this is also included in the 1,000 Genomes Project now. How many SNPs? I told you, using the haplotype tagging SNPs, you can reduce the number of SNPs that you have to test in order to systematically look for association across a certain gene region, for example, or across the whole genome. So if you want to systematically test most of the variation of the genome, how many of these SNPs would you have to test? How many of these 12 million or so SNPs in total? And it's about 300,000 of these tagging SNPs that define most of the SNP variation that is there in the population. And so if you genotype those 300,000 across the genome, you have a pretty complete view of the genetic variation. You pretty comprehensively test most of the variation that is there in your population. So another thing is, so what I have not mentioned before is the term linkage disequilibrium. So you know this picture here. It's basically, I told you that if you have one SNP here, for example, it can predict the SNP allele at a neighboring SNP. So these SNPs are sort of connected, and that is what we describe with the term linkage disequilibrium, or LD. So many SNPs within a block, within a haplotype block, are in LD, in linkage disequilibrium. That is, so it has an advantage because it reduces the numbers of SNPs that you have to genotype to systematically test the haplotypes that are present in the population. It has a disadvantage because once you find association with one SNP, you never know, is this SNP the causative SNP? Let's say you find association with this particular SNP, but it could be one of the neighboring SNPs that are in linkage disequilibrium. And one of these neighboring SNPs, for example, is the one that has a functional effect that is really associated with the imaging trait or with the disease. And so depending on the location of the SNP, that can have different functional consequences. So, of course, it's valuable to know if you have a SNP association, where is this SNP located? Let's look at the regulatory regions. So you know that genes are transcribed, and there is a primary transcript. It has a start and it has a stop codon, and there are introns that are later spliced out, but there are also structures such as the 5' and 3' untranslated regions. These regions are not translated. They are not translated into protein, but they are important for regulation of translation. So if you have a SNP, for example, in the 5' or 3' untranslated region, then it may have an influence on the stability of the RNA or on the production of protein afterwards in a quantitative way. So then, of course, you can have SNPs. Your SNPs may be located in introns, for example, where they may influence gene expression or splicing. Then the other is the SNPs are located in exons. There they may have a direct influence on protein structure or function in the sense of missense, nonsense frame shift mutations. Now, what people have been doing in the last, let's say, 10 years, if they wanted to look for association of SNPs with brain imaging phenotypes or diseases, they have done genome-wide association studies. That is basically systematically testing haplotype tagging SNPs across the genome. And one can do that using SNP arrays. And one of these SNP arrays is shown here. It's by the company Illumina. And this is how it looks like. And there are fields here that you can see. They are made up of millions of microscopically visible beads that are coated with DNA. And what happens if you want to genotype now an individual for several hundred thousands of SNPs, then you can use these chips. And this is just an example for the DNA of an individual that you want to genotype. You have here the genomic DNA. There are 3 SNPs in that individual here in that part. And the first is what you do is you amplify, your whole genome amplify your DNA, and then you break it into pieces, and then you hybridize it to the chip. And the DNA of the individual will bind to a specific position on the SNP, and there it will be made visible by a chemical process which genotype the individual has. Just briefly, so these are these little beads that are on the chip, and the individual's DNA binds to these beads, and then you can, by a chemical process, visualize what genotype the individual has, and that is visualized by certain dyes, by a color reaction. And for example, if the individual is homozygous for SNP1, it will produce a certain color, like here red, or if it's homozygous at the next SNP position for another allele, it would give a green color, and if it is heterozygous, it would have both colors, green and red, and it would produce the mixed color of yellow. So it's basically always homozygous is red or green, and the mixed color yellow is always heterozygous. And this shows you how it looks like. This is genotyping of an individual. You see the picture on the chip, and you see there are thousands of dots in that small region already, and there's green, red, and yellow dots, and the machines automatically read all these dots, and they give you the genotypes for that particular individual in a list. So this is how you can produce in a relatively short time hundreds and thousands of genotypes for a certain individual and for many individuals in parallel. So let's look at other types of variation. SNP variation is the most abundant form, but then we also have microsatellites, mini-satellites. They involve more than one base, 2 up to 1,000 bases, and it's basically a repetition of motifs. Like here, it's a 2-allele motif that is repeated a different number of times in the population. So, and you can have many alleles there. So it's defined by the number of repeats that you have. Larger motifs that are repeated are visible in mini-satellites, for example. These are other types of variation. And one prominent example for that is a repeat polymorphism in the serotonin transporter. This has served for association study with many different brain imaging traits and also disease traits. And what you have is a repeat in the promoter region of the serotonin transporter. And depending on the repeat that you have, the short allele or the long allele, you have less or more production of serotonin transporter. And so you have more or less serotonin transporter depending on your genotype, and that has an influence on the phenotype, or can have an influence on the phenotype. Another interesting and important class of variation are the copy number variants, or CNVs. They are pieces of DNA that are duplicated or deleted, and they involve more than 1,000 base pairs. So basically what you have here is a sequence, a normal sequence. You can have a deletion of more than 1,000 pairs on certain chromosomes in the population, or you can have a certain part duplicated. And so you can have different genotypes for these deletions and duplications. So either here, no deletion, homozygous, no deletion and deletion, heterozygous, and deletion, homozygous. And the same for duplications. And the nice thing is, and that's why these CNVs have been studied quite extensively, is that they are visible on these SNP chips. So this is a picture of the SNP chips where across one chromosome thousands of SNPs have been tested, and each of these dots represents a SNP. And if you have a certain area where you have a deletion, for example, then you get a drop in the signal at that particular position. So here on that chromosome, there would be a microdeletion. So using these SNP chips, you can test whether in particular areas you have deletions or duplications. And then, of course, it's important to know where is the CNV that you are looking at located in terms of functionality. So a SNP may involve a complete gene, so it may have influence on the gene dosage. It may interrupt the gene by basically occurring within a gene and leaving part of the gene there, and the other is part of the gene is deleted, or it can lead to gene fusions even. So it may have interesting functional consequences. So the last part, class of variation that I would like to touch on is the rare variation. So what people have been doing in the past few years mainly is looking at common SNPs, and looked for association of these common variants with brain imaging phenotypes or diseases. So we will later hear about the ENIGMA study, which is a prominent example for SNP association testing for a brain imaging phenotype. And then people have tested these CNVs because they were readily testable using these SNP chips. That was also very convenient. But we know there's much more variation that we can test, and that probably has an influence on brain imaging phenotypes. And that is the class of low-frequency variants that can only be determined by doing sequencing. So they are not contained on the SNP chips. They are too rare to be on the SNP chips, and they often involved only single bases or a few bases, and so that means they are also not visible when you do these conventional CNV studies that are based on SNP arrays. So you really have to sequence. And there are new sequencing technologies. You may have heard about that. Next generation sequencing. It's moving now to third generation sequencing. So sequencing becomes faster and faster by the month, actually, and the costs go down. So we expect that we will soon be able to sequence most of the individuals or patients that we are interested in using these new sequencing technologies. And this is just to give you a flavor how that looks like. This is an image of a sequencing reaction. So in real time, you sequence at many DNA strands of an individual. So each of the dots here is a DNA strand of this particular individual. And the 4 bases, they all carry different dyes, and you basically in real time test what base is incorporated as a next base. So that's real time sequencing basically, and you see you add bases, and then you see where the base is incorporated, and the color of the base that is incorporated will show up as a differently colored dot. So this cannot be done manually. Obviously, machines are necessary to analyze the sequencing reactions here. So it's all in a very small scale, as you can see. And so rare variation is clearly interesting, no doubt about that, but there's also many problems some of these problems we know from SNPs already. So we know there are millions of SNPs in the genome, but only a fraction of these SNPs have functional consequences and are maybe interesting for diseases and brain imaging traits. But it's not easy to filter out which SNPs are the functional ones, which are neutral ones, and at the moment people are trying to find out which are the SNPs that are really functionally significant, that are interesting in terms of function, and the same is true for rare variants. So we have a lot of rare variation in the genome. If you sequence your genome, you will find many very rare variants there, and you have no idea just from the sequence whether they have a functional consequence or not. So just to give you a flavor, there are about 100, if you sequence one individual, you find 200 rare missense variants. That means that lead to a amino acid exchange. Or you find about 100 loss of function variants, and 20 of these loss of function variants. That means there is a truncated protein, or the protein is not formed because of that exchange, base exchange. And 20 of these will be rare or even private, but does not necessarily say that these have to do something with the trait you are studying. So you have to be very careful. You find particular things, but you don't know about the functional consequences of these. So this is something that has to be tested in independent functional studies. And then the last thing I wanted to mention is what people are now also interested in more and more is methylation studies. So you know that the cytosine, the base cytosine in the genome can be methylated or not. This methylation often occurs in so-called CPG islands that are located before a gene, and they have an important influence on gene regulation. So whether they are methylated or not, that has an influence on the expression of the gene. And people want to know, for example, there are individual differences. In one individual, that CPG island is methylated, in the other it is not. And this is something that you want to find out. And you don't see that if you do conventional sequencing. So what you have to do is to treat your DNA in a very special way so that you can chemically transform, you can chemically distinguish a normal C from a methylated C. And then you can sequence that. And then so by doing this, it's a bisulfite sequencing, as we call it, and by this you can really make these differences in methylation of the Cs visible. And this is something that is obviously becoming increasingly interesting also, this look at methylation. Now, and with this, let me wrap up. So I hope I could show you that the genome is highly variable. And so the major focus so far has been on SNP variant, on common SNP variants, and on large structural variants, because these are very easily testable using SNP arrays. And the field is now moving more to whole genome sequencing, which allows also the detection of rare SNPs or single nucleotide variants and small CNVs and indels. And then what is crucial now, obviously, is the functional annotation of the identified genetic variants, and because this will be a crucial step to interpret your association findings afterwards, and also to find out maybe the variants that are more interesting for association studies than others, because that's another problem that you will certainly hear about a lot in the following talks, the problem of multiple testing, because if we're doing these systematic studies, we have a huge amount of multiple testing. We are testing hundreds, thousands, millions of SNPs, and then we have to correct for all the tests that we make, and it would be actually nicer if you had only subsets of genetic variants that you can test where you know they have a functional influence. But to find out whether they have or not, that is really something that is very difficult. It's very time consuming. People are doing that at the moment, but it has not been done systematically for all the variants that are known. And with this, I would like to conclude. Thanks for your attention. Happy to take questions. Okay. Thanks very much.